वेलकम बैक लिसनर. शो का नाम है इनोवेटिव साइंस और हमारी गेस्ट मिस अरीज शेख हमारे साथ मौजूद हैं. एंड इन दिस सेगमेंट वी विल बी टॉकिंग अबाउट how Zabist is facilitating the entire process of your final year project, also known as the research report, जो हर बायोसाइंसेस और बायोटेक्नोलॉजी के स्टूडेंट को लिखनी होती है. तो straight up asking for a direct question, who is supervising you and how do you select a supervisor?
Okay, so our supervisor is Dr. Saika Tabassum. I think everyone in biosciences department has studied a course from her.
And she definitely loves her.
Yes, everyone loves her because she's incredible. Honestly,
right.
I can't praise her enough. And the second part of your question, okay, so you select a supervisor based on their domain.
All right.
So your area of interest for your research has to align with their special, like whatever domain that they specialize in. So for us, Dr. Saika is basically in biochemistry and neuroscience. And nanotech kind of lies in biochemistry.
All right.
So that's how we decided her, and also because we love her, so we thought, okay.
All right. So it's like all three of you love Dr. Saika and you decided कि भाई we want to do our project with her. या फिर it was just like कि हाँ भाई इस पे करना है तो Dr. Saika किसी ने agree किया होगा किसी ने नहीं किया होगा.
No, acha, there was no going back on this decision. There was no back and forth. Is at least idea. I would say that we have gone back and forth a lot. Up until like a month and a half ago, we hadn't had our we hadn't finalized our project.
All right.
So we were like, yeah, maybe change kar sakte hain. Should we change it? Should we change it to this? Should we change it to that? But at the end, we decided that this is something that we like. It's doable, and we're gonna have a lot of fun doing this. So that's why we decided it. And with the supervisor, because I mean that was something that we decided initially. We did not at all think for a minute that maybe we should pick someone else.
Okay. तो लाइक सिंस डॉक्टर साइका जितना आपको पसंद है मुझे भी उतनी ही पसंद है. सो लाइक लेट्स से द इंटायर क्लास वांट्स टू बी सुपरवाइज्ड बाय डॉक्टर साइका और सब कोई ना कोई न्यूरोलॉजी न्यूरोकेमिस्ट्री बायोकेमिस्ट्री के टॉपिक्स ले आए तो शी गोना सुपरवाइज एवरीबडी या क्या हिसाब होता है उसका?
So it's not it's kind of first come first serve in a way, because a supervisor is only allowed to have 2 groups.
All right.
Because and with Dr. Saika, she already has master's students and PhD students, so she can't supervise a whole lot. Every supervisor does 2 groups.
All right.
And that is the limit. After that, then we look so for example, if I'm not saying that this is going to happen, but if a group wanted a supervisor in a particular domain, but they weren't able to get them in time, and then that supervisor had already signed other groups and they were like, okay, I've agreed to actually supervise their projects. So what happens is Dr. Ahmad will usually suggest another supervisor who does not have that many groups.
All right. So you were there right on time, just getting her and getting it signed by her. Oh, well, pretty good job with that. I'm going to take notes for that.
We were lucky, I guess. I mean, it's just a matter of luck. I can't say anything else.
All right. तो जितना आपने काम किया है, since you said you made the extract and everything, तो ये सब आपको खुद आता था और it was a supervisor who was telling you कि अच्छा भाई बेटा अब इस तरह करना है और अब इस तरह करना है, अच्छा ये इस तरह नहीं इस तरह करना है. What is the role of the supervisor in your project? Is it like she helps you throughout the step or she just looks over कि हाँ सही है या गलत है?
That's actually a really good question, and the thing is it varies. So depending on your project difficulty level and your familiarity with the topic that you're actually doing, so a lot of people started, so for us, for example, we're doing nanotech that we hadn't studied before.
All right.
But in terms of methods, what we're doing in the lab, we have to bring those methods ourselves. And then our teacher is going to approve them. So your supervisor is there to literally just supervise you. She's not going to control everything or guide you in that sense that she's basically dictating every single instruction. Like she's telling you, okay, now do this, now add this, now add that. It's more or less your thing. I mean, you have the idea, she approves it, then you come up with the methods and then she approves them with proper references, of course.
All right. So my next question to you is, since you have not studied nanotechnology, अब तक आपने पढ़ी नहीं है, तो how did you come up with all the methodologies that you will be using? What is the thought process behind it? Do you have to do a certain thing? Obviously books पढ़नी पड़ती होंगी, but how does that go?
So the general rule that we should follow, especially as undergraduate students, is take reference papers
All right.
I mean, don't create your own methods.
Okay.
Yeah.
The
so
students
just-
do that?
they do, and like when you're appearing in front of all the faculty members for your title defense, you will realize that it's been a huge mistake, because they will grill you and they will ask you why you picked this method, and you need to have solid references. So what happens is, if you're making, for us, we picked polite, and we were making nanoparticles from that. And as I mentioned before, there wasn't a lot of research for polite, so what that meant was we had very limited research papers.
All right.
So without those limited research papers, we would have been completely grilled. It basically adds authenticity to your project, because we're not qualified to come up with our own methodology.
Yeah, we're just undergraduate students.
Exactly.
All right. तो जो भी आप अपना experiment कर रही हैं using pulai, so where are you getting that pulai from? Is Zabis getting it for you? Are you growing it over here? क्योंकि Zabis में भी एक मैंने सुना था project EPC ने ही लगाया था, vertical garden something, तो पता नहीं शायद उसमें हो pulai. And what about all the equipment that you're using in the laboratory? सब available है, all the chemicals, you know, financially Zabis कितना support करता है?
Okay, so depends on your project. If you're sourcing things that aren't available here, you have to contribute for that yourself. You can't really Zabis does not compensate you for that.
Okay.
So yeah, we basically, so for plants there is a specific process that you have to follow. You have to get your sample and then get it identified from KU Herbarium, and they give you a serial number that indicates that yes, this is in fact the species that you wanted, or whatever species that you're calling it. So for us, we got an ID number that basically tells us that yes, this is Acacia modesta, whatever.
What is it? Can you please repeat that again?
Acacia modesta, that is Pulai's scientific name.
Alright.
So whenever we're going to the herbarium, they don't just say Pulai, they use the scientific name.
Alright.
And after that, I mean, for us, we were really lucky, because when we went to the herbarium, the person that was there, he basically helped source a lot of it, because we had a very limited amount of pulai. So as you know, the flood has affected this, most of like the places that pulai is grown in, so we ran into a lot of trouble sourcing that, and that's why we were considering changing our project, because we were like, if we're sourcing plants and the plant isn't available, it might cause a lot of problems.
True.
But luckily, because of the person at KU who helped us a lot, he basically helped source the plant material. So yeah.
And who paid for it?
We did.
Oh my god.
Yeah, we pay for it ourselves because it is our project, and it's like that.
Okay, so what about the silver nitrate? Is it available in the laboratory here?
Okay, so silver nitrate is something that you have to make, but we do have the raw materials that we need to make it.
Alright. And how do you make silver nitrate?
Okay. I don't know how to explain this. I mean, this is very technical, but it's basically you take silver
and you take nitrate and you mix
it together.
Okay.
And then you get silver nitrate.
Okay, what about the rest of the equipment? Like, you know, there's a lot of laboratory apparatus that you're supposed to use. So how do you go about it?
It's available. We have 2 labs. We have 154 Valley Lab and then 173 Light Lab. And both of them have pretty much all the equipment that you need. Sometimes if another group is working, or if multiple groups are working in the same slot, then maybe you might run out of some. Like for example, today we needed some beakers, so we had to go to the 154 lab, get those beakers, sign off on the sheet, and then write down what we were taking from there, and then bring it back to 173. So yeah.
Okay, so like all the students rush into the labs together.
I mean, when you're in your 7th semester, we have our classes together, right? So usually people have the same spots that are free, the same time slots that are free. So today we were there after our bioinformatics class. We were all there in the lab, literally like 8 groups or something were there working in that lab.
Oh my god.
So yeah.
So that's a lot of crowd, but
it is.
I hope that doesn't affect, have you ever been in a fight with somebody working together?
I mean, not really. Most people are nice, and we have like a very lively banter going on with the class. Like everyone is just teasing everyone, and everyone's just having a good time. So even though we were there for like 3 hours, and my friends are still there.
Okay.
So the whole banter thing, like the whole, back and forth with your friends makes it a lot easier. It's not just, boring work for hours and hours and hours, because it does take time, especially when you're an undergraduate. You don't have that much experience. You can't just, do everything very quickly. You can't make plant extracts that quickly. We're about to start the synthesis of nanoparticles, and that's going to take a lot of time.
Okay.
So, I mean, it's nice to have them there, to be honest.
Have you ever been scolded for just having fun around in the lab by your supervisor or the lab technician?
Okay, so we weren't scolded. I mean, we were technically, but so two of the supervisors were there. Miss Neha and Miss Lusa were there, and then I think one of the faculty members just came in to see what was going on in the lab, why it was so noisy, and then our teachers were like, please kids, please, a little bit lower, just a little bit lower.
Okay, so I can totally understand where there's a bunch of girls in a room, there's no way you can keep quiet.
Yeah.
Okay, there was an interesting terminology that you mentioned, title defense. So we're going to talk about that. That seems very interesting, but after this short little break.